So hello and welcome to week 10 of the Next Generation Sequencing class. This is the beginning of the lectures that I'll be giving on RNA sequence analysis. So this is our favorite way in Next Generation Sequencing analysis of quantifying gene expression. And there's a lot of applications, some of which I'll be covering today, and some of them you'll be dealing with in your homework. In particular, differential expression analysis is probably the most common form of RNA-seq analysis that's done today, in which we try to quantify differences in gene expression between different biological conditions. So I'd just like to begin by giving some background on transcription, some of the biology, since I know that there are people in the class who are coming from a computer science background and others that are more comfortable with the biology. But just to give some introduction, I'd like to talk with you primarily from a eukaryotic perspective, including humans. And in eukaryotes, as you all know, transcription of genes is the means by which the sequence of the DNA is converted into an mRNA and eventually translated into a protein. And the process of transcription involves the splicing, the production of what we call a premature mRNA or a nascent mRNA, which is then spliced to remove the introns, from what's called primary transcript here. That splicing or removal of the introns leads to what we call the mature mRNA. Of course, there's other things that take place here, where you have capping and the 5' end and a poly-A adenylation of the 3' end. And then keep in mind that only certain parts of this mature mRNA are actually translated into a protein, and that part we call the CDS. And that becomes the template that a ribosome uses to build a protein sequence from amino acids. So this process of splicing in eukaryotes can be quite complex, and it leads to a phenomenon that we call alternative splicing in eukaryote genomes. That is where different transcripts or mature transcripts can be derived from the same exact gene. So this is a difference in exon usage, from the same original gene. So here in this example, this particular gene has 5 potential exons, but with differential splicing, an exon can actually be removed as if it's an intron, for a certain transcript. So here is a complete complement of exons for this particular transcript. We'll call it protein A. So this transcript, when it's translated, leads to protein A. And notice it has 1, 2, 3, 4, 5 transcripts, where this transcript here has 1, 2, 3, 4, and 5 exons. So all 5 exons found in the original gene, whereas this alternative splice variant has 1, 2, 4, and 5. So exon 3 is spliced out, and here exon 4 is spliced out. So there's a lot of things that can vary in alternative splice variant. The 5' and 3' ends can vary. The exon content, as illustrated here, can vary, and there can be altered translation starts positions. So what this does is we have what we call gene models, where we predict the intron-exon structure of a gene, and alternative splicing means that we need to have more than one model per gene. So in the human transcriptome, there's roughly 20,000 protein coding genes and greater than or more than 100,000 distinct transcripts. So on average, each gene has multiple alternative splice variants. So another thing about eukaryotic transcription is that we're not just talking about the transcription of protein coding genes, but there's a whole array of known RNA species in the eukaryotic cell. So what we talk about frequently when we're talking about genes is the message or messenger RNA, but other classes of RNA we call non-coding RNAs. Historically, these are the two most common that are discussed in the literature prior to roughly the late 1990s, ribosomal RNA and tRNAs. So rRNAs and tRNAs, the ribosomal RNAs are used for essentially making up the ribosomes. The tRNAs are used for building the amino acid sequences into growing proteins. The small RNAs, their functions are far more diverse and frequently involved in various forms of gene expression themselves, including antisense RNAs and RNAs that bind to the 5' and 3' tails of mRNAs and facilitate their breakdown or their preservation in the cell, and are used for a variety of different and sometimes obscure gene regulatory mechanisms. So sometimes we call these microRNAs or short non-coding RNAs. There's also now known long non-coding RNAs that can be, for example, an antisense RNA that complementary base pairs the mRNAs themselves, or there can be long regions of non-coding intergenic DNA that's expressed as long non-coding RNAs, and their functions are now only beginning to be understood. Now, one of the things that's important in RNA-seq analysis is to first be aware that all of these RNAs exist. The second thing is to be aware that by far the most abundant class, is the ribosomal RNA. So up to 70 to 90% of a cell's ribonucleic acid content, the RNA content can be ribosomal RNA. And I'll also point out here that this is probably the RNA that people are least interested in because it's being derived from only a handful of 3 or 4 genes in the cell. And so really this is for differential expression analysis and for discovery of transcripts, which are two of the major applications of RNA-seq. We're totally uninterested in this class of molecules. And so, as I'll point out later, researchers go to great length to removing ribosomal RNA from their library preparations prior to the RNA sequencing process. And so you should be aware, however, that ribosomal RNA contamination at the bioinformatics step is a real problem in many libraries, and so you need to be aware of this issue. So the human transcriptome, this as of April of this year, contains a total number of genes of 57,000. Notice that this is different from the number I cite here of protein coding genes. So people call tRNAs, rRNAs, other small or microRNA coding loci are referred to as genes, but we don't frequently call them, obviously we don't call them protein coding. And this just gives you some summaries of the different types of RNA that have been discovered in what's called the GenCode project, which is part of the greater ENCODE project, which is to understand or characterize the function of virtually every position in the human genome. So the ENCODE project, I encourage you to read further on simply because it's a fascinating attempt to characterize the full breadth of function in the human genome. The GenCode project is sort of a subset of that project whose aim was to characterize all of the transcribed regions in the genome. So I thought I'd give you some picture of some organismal differences in gene sort of organization and gene expression. So in yeast, if someday you end up working on yeast, you'll notice that yeast typically only have one transcript per gene. So you don't see a lot of this alternative splicing. In fact, there's minimal alternative splicing. Instead, you see other complicating factors like genes tend to overlap. And you also will notice that genes are very tightly packed in the yeast genome. So here's an image from the G-browse for yeast. And what you can see here is these models here. I should have put for contrast one that had an intron in them, but essentially all of these gene models are single exon gene models. So there are no genes in this particular image with introns. Okay, and that's very different from the vast majority of eukaryotes. So when you have single exons, essentially alternative splicing is impossible. So I thought I'd also introduce a classic example of alternative splicing in humans as it pertains to disease. So we've talked a little bit about cystic fibrosis in the class and the genetic basis for the disease. But one of the reasons why individuals end up having the disease is because of mutations at individual positions in an intron that lead to failure to alternatively or to correctly splice in any event the gene called the cystic fibrosis transmembrane conductance regulator. So this is an ion transport that's transported or expressed in the lung tissue, and exon 9 is the problematic exon. So when exon 9 is included in the transcript, okay, you end up with a normal tissue. However, when exon 9 is skipped via an alternative splice event, this is what leads to a cystic fibrosis. So the ultimate reason for it is because of a mutation. TG is a short tandem repeat of TGs in the intron. So TG run of 9, TG run of 10, run of 11 TGs in a row, combined with a run of 9, 7, and 5 Ts leads to the normal phenotype. However, these variable numbers of runs of TGs and Ts end up leading to failure to incorporate the exon 9 into the normal mature mRNA at this gene. So it's not that these types of things are unusual or foreign. It turns out that when you look at the total spectrum of disease-causing mutations in humans, that roughly 15% of them are mutations that affect splicing. So all of this to say that essentially from the biology of the cell, the ways in which genes are alternatively spliced turns out to be really important, and hence the need in part for discovering the full complement of alternative splice variants in the cell. So I just thought I'd throw here an example of probably the most extreme example of alternative splicing that's known in eukaryotes. It's the DSCAM1 gene, where there's up to 38,000 types of transcripts. Essentially, there's these runs of potential exon 4s, exon 6s, exon 9s, and exon 17s in this particular gene. And what in each cell can happen is that there's a choice made between which of these small red exons here are actually represented as exon 4 in the mature mRNA. Same for 6, 9, and 17, and you can end up with any and all combinations of exons for 4, 6, 9, and 17 in that mature transcript. Okay, so each one of these small vertical bars here is a different potential exon that gets placed, in this case, for exon 4, exon 6, exon 9, and exon 17 in this mature mRNA. So this essentially is a way of obtaining extraordinary functional diversity in Drosophila melanogaster, or the common fruit flies, neuron tissues, such that this protein can be slightly differently expressed and given in different cells, giving each cell a very distinct signature on its cell surface for this particular type of protein. So I thought I'd give a little bit of historical background, especially for the non-biologists or those of you who are new to the field of genomics, because it provides some context and some, I think, useful differences between historical methods of analyzing whole genome gene expression data versus RNA-seq. So historically, I'll really only key on a couple of these. ESTs are still used for discovering the gene models. So this is how we frequently in the past have discovered novel genes and their exon content. And essentially this is done by creating cDNA or complementary DNA. This is taking an RNA from the cell, making it into DNA so that you can clone it into a library and sequence it. So this is the earliest way of sequencing mRNAs. And so EST is expressed sequence tag. And you'll frequently still see these in gBrowse images where you'll see the gene models that are supported by various ESTs. So remember that an EST can confirm the presence of a particular exon in a particular mature transcript because the introns will be spliced out. I should mention, for those of you who are not the biologists in the class, that the introns that we've been talking about here, that the pieces that are getting removed are removed so quickly in the following the initial transcription process that at equilibrium there's virtually none of this type of molecule, this primary transcript in the cell. So when we sequence both RNA-seq or we sequence ESTs, we almost never detect the introns. Although when sequencing at such great depth, with RNA-seq, as we'll describe, you do start to pick up some introns in the sequencing, although at very low level. So, so that's EST's expression microarrays. I'll describe in a minute. This is another, for a long time, dating back to the mid to late 90s, this was the method of choice for whole genome analysis of RNA, particularly mRNAs. So here's just a quick refresher on cDNAs and express sequence tags. We clone the amplified DNA into or we clone the cDNA into a vector. It's not amplified yet. We simply, to make the cDNA, we prime either with a hexamer or a poly-A priming. This is using actually a poly-T that binds to that run of adenines at the end of most eukaryotic mRNAs. You prime the synthesis of the second strand, or a DNA strand that's now complementary to the RNA. You then make it double-stranded and then clone it into a vector, and then use bacteria to grow that, or to replicate that inserted molecule together with the rest of the plasmid. Then you use primers that are specific to the plasmid, because you don't obviously know the insert or the cDNA that you're interested in. You don't know its sequence and you don't have primers for it, so you use primers sitting in the plasmid here and sequence across the EST to discover the various exons that are in that transcript. So they're typically very short. They're there to verify the transcript presence and absence, and also to identify the exon content of the transcript. But generally we don't use this for quantification, although I have seen it done simply because we don't have enough of these to actually quantify. Although if you sequence several millions of these, you can begin to at least identify the very most abundant transcripts in the cell, because you'll continue to sequence those transcripts. So microarrays, this is the classic technique for detecting differential expression, whereas ESTs are primarily used to discover novel genes, or to discover the various alternatively spliced transcripts and the exon content of particular mRNAs. Gene expression or differential analysis, where we're trying to, say, measure the difference in transcription at each gene locus in the genome between two either cell types or between two treatments, where we've treated two different samples to different environmental conditions and want to see how the cell responds. These are sort of classic differential expression experimental designs. So we start with 2 mRNA samples, say from cancer, say from normal cells. In the cDNA synthesis step, we incorporate fluorescently labeled dyes into the nucleotides that go into making the cDNA. These are in this, typically referred to as red or green. Technically, they're called Cy3 and Cy5 labels. We then competitively hybridize those to a microarray. So a microarray is just a glass slide that has individual little spots that contain, tens of thousands of probes representing a single short DNA sequence. So each sort of spot is a clone of itself, where you have many probes that represent the exact same DNA sequence. We then competitively hybridize the Cy3 and Cy5 cDNAs to the entire microarray, and we we create an image of the microarray in which we can quantify the relative amount of green and red dye at each of these many tens of thousands, if not hundreds of thousands of spots on the microarray. And in that way, we can contrast the two colors and get a relative measure of gene expression between our 2 original cancer cells across probes that represent the vast majority of the exon sequence in the genome. I'll just point out really quickly that this constrains your approach, or your ability to discover new genes, for example, or to measure the relative expression of genes that are unknown, because you're having to start with a microarray that already has probes from genes that are known. Okay, so let's talk about RNA-seq and a little bit about how it works. In this particular example is restricted, mostly to mRNA. That, of course, is for many researchers what we're primarily interested in, because it's the protein coding genes of the cell. However, there's a huge, effort to understand these small RNAs, these microRNAs, because their role in the cell is certainly essential in many cases and probably related to disease in others. And so understanding both is obviously important, but for simplicity here, we'll restrict our conversation to mostly to mRNAs unless I mention otherwise. So essentially, we start with with RNAs. We then either fragment the RNA itself, or and then prime it with what we call these random hexamers to make cDNA, or we poly-T prime the cDNA and then fragment it before making our libraries. This is by far the approach that's adopted now. As I'll show you in a minute, there's some biases in doing it the other way, where you first make the cDNA, then fragment the cDNA. So what we tend to do is fragment the RNA first. We make our cDNA from these small RNA fragments using these random hexamers to prime the synthesis of cDNA. We then attach the library adapters, to the ends of these cDNA molecules, and then we go forward with sequencing. I should mention that we frequently use paired-end sequencing of these small cDNA fragments, although it certainly initially anyways was far more common to see single-end sequencing of these cDNAs, simply because all you really need is one count or one read to represent the initial presence of a single mRNA in your sample. And so the idea was, why do you need to sequence both ends if the point is just to count the number of mRNAs in the cell, so that you can contrast them, say, with the number of that same mRNA in, say, a different sample. However, paired-end sequencing offers a lot of advantages. One of obvious one is that you can tell for reads that don't span 2 exons. Say you have a read that maps to an exon, its mate may map to another exon, and that would tell you something about the exact splice variant that both reads came from. Okay, so we sequence the general strategy is to sequence these using short read sequencing. We sequence these individual small cDNAs that each individually represent an original mRNA molecule. We then map them back to the genome. First pass, as I'll describe, to exons. We then use what we call splice read aligners, which will break the reads up into fragments to identify those that don't map, to see if we can get them to map to alternate sides of potential introns. And then we look at the expression profile across an individual gene to look to see the number of reads that map to the different positions in the gene. That tells us which exons are present, and also provides us with a means of contrasting the abundance of reads in one sample compared to another sample, or potentially between genes in the genome. So sometimes we're interested in which are the highest and lowest expressed genes in the genome, and this data can also provide us with that information. So RNA-seq works. That's the point of this slide. This is the Mortasavi paper that I assigned to you for reading in the class. Is really the first study to use RNA-seq and to introduce what we call the reads per kilobase of transcript per million reads of the library, a measure of read abundance or counts. So, these are technical replicates here. We'll be going into experimental design in the next lecture, but technical replicates, while they're not common anymore in RNA-seq because it's expensive, you can see that the technical replication is good. So just the same tissue exactly prepared as separate libraries and then sequenced separately or independently on a short read sequencer, and you get, the read counts, where each point represents a single gene, from on the x-axis technical replicate 1 and on the y-axis technical replicate 2, and you get a strong correlation. The sensitivity and dynamic range, this is something that differs dramatically between microarrays and RNA-seq data, is that the dynamic range is over 5 orders of magnitude here, where you have linear relationship between the read counts and the number of transcripts in 100 nanograms of mRNA, quantified by much better techniques even than RNA-seq, using a real-time PCR. So I didn't mention real-time PCR in my introduction, perhaps I should have, but real-time PCR is a way in which is the gold standard for quantifying transcript abundance in molecular biology, but it's typically only done for a few tens of hundreds of genes, and not typically for entire genomes or transcriptomes because of the expense and time associated with it. So as we've talked with DNA sequencing, the RNA sequencing library construction methods are very important, and it's important for you, the bioinformatician, to know something about the techniques that are used in the wet laboratory because it informs how you conduct your analysis of the RNA-seq data. So we begin. I've highlighted here in red the parts that I think are particularly important for the bioinformatician. But we start with RNA extraction. We get rid of all the DNA by essentially degrading it with an enzyme that degrades DNA but leaves RNA intact. Okay, then this is the step that's important. I mentioned the contamination of ribosomal RNA. There's two techniques. You can either deplete the ribosomal RNA by competitively binding it away using ribosomal probes. That's called ribodepletion. Or you could say bind all of the mRNA to a column that has poly-T probes so that the mRNA binds, and then you let the ribosomal RNA wash through the column, and then you elute the enriched fraction for mRNA. So all that to say that there's two ways of getting rid of the ribosomal RNA. They are essential, and as a bioinformatician, you start seeing reads mapping to ribosomal RNAs, and you might want to ask the person who prepared the libraries, how they did it, and you could talk to them about how effective they were. But ultimately, you don't want to be sequencing ribosomal DNA or the DNA produced from ribosomal RNA because it's uninteresting for most biologists. We're not particularly interested in the differential expression of the most highly, transcribed genes in the genome that, mostly we don't think are likely to contribute to differences in phenotypes or differences in responses of cell types to certain environmental stimuli. Okay, so we fragment the RNA. That's just necessary for the short read sequencing. Remember that we need small, 2 to 5 or 600 base pair fragments to get the clustering to effectively on the flow cells and to get the sequencing accomplished. Of course, we need to create cDNA because we can't directly sequence RNA. We always first have to convert the RNA to cDNA before sequencing for biochemical reasons. I wanted to mention here, in one of the Wang paper said that this was an older paper now, back in 2009, I believe the Wang paper is that I assigned to you, but they did mention strand-specific library construction as being something of the future. Is currently very difficult to do. Well, this is something we now do routinely. Okay, what this allows you to do is to detect which strand of the genome did the plus or minus strand, did the transcript actually come from? Okay, so remember now that a gene typically, we think of the gene, the mRNA is coming from the sense strand. That's either the plus or the minus strand, because genes can be either oriented on the plus strand or on the minus strand. However, you'd want to know which of the two strands is actually being transcribed. Okay, so generally a gene will be transcribed on its sense strand, but we now know that there's lots of antisense RNAs out there that are transcribed, and so this allows you to tag which strand the particular mRNA is coming from. We also have to do PCR, and this again is a reason for getting enough, we do this to get enough density to facilitate the cluster formation on the flow cell. But obviously you don't want to do a lot of PCR because you're trying to, the counting procedure that we use to count the mRNAs is simply to count up the reads that map to an RNA, and we assume that each read represents a separate mRNA molecule. However, a read, a pair of reads that, both are coming from the same PCR reaction could actually come from the same mRNA molecule, thus double counting, the number of mRNA molecules. If we were to, sequence the same PCR product multiple times, we'd be, sequencing the same mRNA sample or mRNA molecule multiple times, which is actually not the goal. So this becomes a bit of a problem in cases where you have biases where particular transcripts are, more efficiently PCR amplified than other transcripts. So it's something that people are aware of, and I think the trend is eventually going to lead away from doing any PCR at all. However, it's still very much necessary to do PCR to seed to get enough DNA to seed the flow cell, to seed the cluster formation on the flow cell. So we do size selection again, we do library quantification, and then we do the short read sequencing. Okay, so it's probably obvious to you that mapping RNA-based reads back to a reference genome is a little bit different than mapping DNA-sequenced reads, and that's because of the splicing phenomenon. So in black, you have pieces of DNA that should map to the reference genome perfectly, whereas in blue you have pieces that are spliced. So there's a piece of DNA in the reference genome that is present in the reference genome that is not present in the transcript. And so in blue you have reads that span those boundaries. So in the first pass, so I'm here referring to Top Hat, which is the spliced aligner that you're using in your homework. In the first pass, you have Bowtie aligning the reads, in which it just aligns all reads the same way it does if you had sequenced with DNA. But then there's a whole, population of unmapped reads. So what Bowtie, or what Top Hat does rather, it first aligns with Bowtie, then it goes through and it looks for the, for reads that if you split them, will map, to exons on either side of a sequence like this. So it splits the reads and then looks carefully to see where those smaller, subreads, if you will, map to the reference in this fashion. Okay, so that's how the spliced read mapping gets done. Try to make sure that you can wrap your head around the fact that the blue reads will not map to the reference genome in a straightforward fashion because they're split by introns, which are spliced out in the molecule that we're sequencing. So as part of an introductory analysis, sort of lecture to RNA-seq, I need to make sure that we at least understand the need for normalization. We can't just count up the number of reads in RNA-seq analysis by itself and not normalize. And that's because just in the trivial example, if we're trying to do a differential expression analysis where we have sample 1 and sample 2, say that one's from a tumor cell, one's from a normal tissue cell, if one library is bigger than the other so that there's more reads, it would look like all of those genes are overexpressed in that one sample compared to the other. Okay, that's just a trivial example of why you have to normalize. You have to take into account the library size. If you think about it, you also need to take into account the length of the gene, because longer genes will have more reads mapping to them than shorter genes, et cetera, et cetera. So we have to normalize. And part of the next few lectures will be in getting into a little bit more in detail on how we go about normalizing and dealing with the statistical issues. But the first attempt at normalizing was done by Mortasavi in that original paper, where they proposed a measure called the RPKM, the reads per kilobase of transcript per million reads. So essentially all this is counting the reads that map to a transcript, and then we're dividing by the length of the transcript and the size of the library, or in millions of reads. Okay, and so this becomes a length of the transcript and total count normalized procedure. So you'll also see reference to the FPKM, which is the fragment per kilobase per million reads, and this is essentially the same concept. It's just that this is really meant for a paired end context. Okay, so for paired end libraries, we don't want to double count a particular fragment, okay, or a particular cluster on a flow cell that produced a pair of reads that represent the same mRNA molecule. Okay, we really would like to only count one end, okay, because that is, represents the number of mRNAs where that fragment came from. So each mRNA in principle is tagged once, but if you have sequenced that mRNA twice, once on either end, then you're doubling your count. So this is necessary, to switch to FPKM, because it avoids double counting. It also avoids undercounting those fragments in which only one read mapped, in which case you might only count a particular transcript once in that case. Okay, so this tightens up or reduces the variance associated with this trivial problem of dealing with paired end data. So this is what sort of count data look like across a cell or gene. This is averaged across 5,000 yeast genes, where you frequently get a loss in red. You get a loss of counts sort of at the 5' and 3' ends of the genes, but you get high read counts in the middle. Blue was using a different library preparation method using cDNA fragmentation. This is just kind of an aside, really. I really just wanted to show you the distribution of RNA based fragments that you get when you do RNA fragmentation. And you get this roughly even distribution across genes. This is a particular gene called SES1, and this again just shows you sort of some normal, perhaps Poisson, but likely a little bit over dispersed compared to Poisson expectations for the sort of the read counts, across an individual gene. Again, showing in blue the RNA fragmentation. The red is the RNA fragmentation in the library preparation protocol. The blue is the cDNA fragmentation. So we generally do RNA fragmentation, and you can see a roughly even distribution of read counts across this particular gene. This is just really quick, maybe some, for those of you who have done some RNA, some microarray analysis in the past, or are familiar with microarrays like many of us have been for a very long time. Just a few things to highlight here. Gene sequences must be known, right? The probes to build your microarray, with the possible exception of tiling microarrays, where you essentially build a probe for every position in the genome, which are very expensive, and only used in humans and a few model organisms. We essentially cannot detect novel transcripts except potentially with the tiling microarrays. Whereas in RNA-seq you can, and you don't need any information about the genome's mRNA content in order to extract information about the presence of genes and transcripts using RNA-seq. Here you can, in RNA-seq you can detect the full array of alternative splice variants, where you're limited again with that in microarray data simply again because you don't have probes representing all pieces of DNA in the genome generally. Here we have a lot of background noise due to the fluorescence intensities, so you always end up with background signal in microarray analysis that has to be corrected for, where we have far less sort of sources of noise in transcript abundance estimates from RNA-seq. One thing that we'll come across is that the signal is unconstrained by the abundance of other mRNAs, so that's something that I left out here. But this is each sort of probe is independent of other probes. And whereas that's not true in RNA-seq, where the abundance of one gene may be constrained by the superabundance, say, of another. So if you have a lot of a particular gene in your library that's overexpressed, that's going to necessarily draw counts away from some lowly expressed genes simply because you only have a finite number of reads in your RNA-seq library. So the statistical sort of ramifications of that are quite important to consider, and we'll do just that in upcoming lectures. There's more variability in microarray data. There's a little less in technical replicates in RNA-seq. Signal background noise, as I mentioned, is higher in microarrays than in RNA-seq. And then importantly is the dynamic range. So we can measure differences in expression for very lowly expressed genes and very highly expressed genes in RNA-seq, where we can't do so at the extremes of very low and very high expression in microRNAs. And that's what I'm referring to as the dynamic range. So just how many different orders of magnitudes in expression can we detect differences between two samples? And we can detect differences in RNA-seq over much greater spread in magnitude than we can in microarrays. So finally, to tie up this introductory lecture, we use RNA-seq in a number of applications. I've told you at length about how we use it to detect alternative splice variants in different forms of transcripts that are in the cell. We use RNA-seq to quantify the expression of different transcripts, okay, so say from different cell types or different treatments or different quantifying how genes respond to different stimuli. We use RNA-seq for that. We use RNA-seq to come up with transcription atlases or transcript atlases. So you'll see like the cancer genome atlas where people are going in for every cancer type trying to determine the full range of transcripts that are present in different types of cancers or at different developmental times in say human embryos or in tissues or different cell types, etc. So this is just generating catalogs of transcript abundance and presence, absence of individual splice variants. Gene fusion detection, this is important in cancer research where we know that transposable elements, movements, a major structural variants happen because of spontaneous mutation, and we can detect novel gene products that are produced and expressed in cancer tissues, and so that's led to many important discoveries now in gene regulation in cancer. Gene discovery, so in non-model organisms where we don't know what part of the genome is expressed, we can obviously reconstruct that very easily with RNA-seq and not only discover new genes but discover various transcription alternative splice variants. And then SNP analysis. So this is one I haven't mentioned, but one I'm discussing a project and considering working on a project with a colleague who's working on very large genomes of plant species where we know nothing about the gene content, nor do we know SNP where there are SNPs in the genome. But obviously, where you'd like to start is in genes. Usually, we'd like to know where there is genetic variation within genes because we think that genes are the most functional or most likely to be functional in the genome. So we'd like to know where there's variation in genes, and so one way to do that is to just sequence RNA and try to call SNPs from RNA-seq data. And we're currently planning experiments to do that in a type of gymnosperm. And then finally, for regulatory network inference, this is something where you can do time series analysis to get whole transcriptome information over very short time spans following some stimulus, and you can use that to use different mathematical models to predict which genes are controlling the expression of downstream genes, whose expression gets kicked in at a later time following a particular stimulus, owing to sort of cascades of transcription factors and other regulatory network phenomena that lead to these downstream expression and changes in expression following some stimulus. So that's something that we have a lot of work going on at NYU to do just that. Okay, so with that, that's the end of this introductory lecture to RNA-seq, and with that, I'll talk to everybody next time.